Now, before we finish talking about action potentials, we have to talk about how they move, because an action potential by itself, just a temporary change of the membrane potential, is not actually going to do anything. In order to be a signal, in order to allow cells to communicate, the action potential has to travel along the axon. And when that happens, we call it action potential conduction. So the movement of the action potential down the axon we call conduction. And it's caused by basically a chain reaction in which the depolarization of the membrane leads to voltage-gated sodium channels opening. And then as those channels let sodium in, that causes more depolarization. That the current that's produced, so as the positive current enters the membrane or enters the axon, it travels down the axon cytoplasm. So the inside of the cell becomes more positive. That causes positive current to travel along the axon, and that then triggers the opening of more sodium channels. So again, next to, let's say if there are sodium channels here in the membrane, there are sodium channels all over the axon here. So there are going to be sodium channels here too. So as you go down the axon, every time some of these sodium channels opens, that causes the adjacent ones to open too. So then if we move down a little bit, a millisecond later, we see that the channels here now are open. That's creating more positive current, which is traveling down the axon, creating more depolarization, causing more channels to open. So it's this chain reaction. It's kind of like a fuse being lit on a bomb. The energy from the burning causes more burning, and that travels along the pathway. And again, this is all happening very, very quickly. So the current moving down the axon is very fast, faster than the action potential itself, and so it's constantly triggering more and more of these channels to open, keeping itself going. Now, the axon itself has no direction to it, so it's not like the movement of the current travels in both directions. So you see the current traveling in both directions, but the action potential actually only travels in one direction. So in other words, even if you were to, or the reason that the action potential does not reverse at any point, doesn't stop and go backwards at any point, is because behind the action potential you have this outward potassium current. So again, you have sodium coming in, and behind the action potential you have potassium going out, and plus the fact that the sodium channels are inactivated. So in this part of the axon, right behind the action potential, you have zero or very little sodium conductance and very high potassium conductance. So that means that even though the current, the positive current travels down the axon the other way, it immediately leaves. So it's not able to trigger the sodium channels that are back here. So that's why the action potential only travels in one direction at a time. But there is no, other than that, there is no directionality to the membrane or to the cytoplasm. So a typical, or most of the time, action potentials are generated at the cell body and move away from the cell body toward the axon terminal. When that happens, we call it orthodromic conduction. Again, that's kind of the typical way that action potentials are propagate, but there's nothing stopping you from generating an action potential that goes away from the axon terminal toward the cell body. That's called antidromic conduction. And both, again, are possible, but orthodromic is much more common. It's the typical way action potentials travel. In fact, antidromic conduction is really only going to happen artificially. So when you're generating an action potential by, say, depolarizing the axon itself and not the cell body. But the velocity of conduction, how long or how quickly the action potential travels, is determined by, again, the ability of that current to flow down the axon. So the cytoplasm inside the axon is where the current actually travels through. The cytoplasm has, lots of ions in it, lots of salts, so it has it's very conductive, compared to pure water. But the limitation on the movement of current through the cytoplasm is current leaking out through the membrane. So again, even though there are very few or fewer open cation channels in the membrane, there are some, and so this positive current does have the ability to leak out. Or you can have negative current or negative ions or electrons coming into the axon. That would have the same effect. So the speed of the action potential does depend on the ability of current to move down the axon, and the more current that can leak out, the slower the action potential. So one of the things that affects the leakage of current is the diameter of the axon. If you have a very thin axon, that means there's a lot of surface area relative to the volume or the diameter of the axon. And so there's lots more space for current to leak out, again, as a percentage of the actual volume of the axon, the cylindrical volume. Meanwhile, if you have a thicker axon, you still can have current leaking out, but the percentage or the surface area of the axon relative to that cylindrical volume is less, and therefore the current leakage will be less. So one of the things that affects action potential conduction velocity is diameter. The thicker axons have faster action potentials than thinner axons on average. In fact, in some situations, some organisms have very, very big axons. So a good example of that is the squid. Squid have, this is basically their nervous system. They have a brain here close to their eyes, and then they have nerves that go out to the muscles and sensory systems of their body, especially this structure called the mantle. So the mantle kind of surrounds all the internal organs of the squid, and it's this big kind of water reservoir too. So one of the ways squid and other cephalopods are able to escape from predators is they can fill this mantle with water and then squeeze muscles that surround it to squirt all that water out the opposite direction, and then they can jet away from their predators very, very quickly. So for that to be useful, though, you have to be able to send the signal from the brain to those muscles very rapidly. So if the squid sees a shark or something that wants to eat it, then it can send a signal along this nerve to those muscles very rapidly. And so these axons that connect the brain to those mantle muscles are what we call squid giant axons. They're very thick. They're almost a millimeter sometimes in diameter, which is big enough to see with the naked eye. And so these are huge axons. And so and that's just one example of an adaptation that have evolved to deal with this problem. Now, in mammals, we do not have giant axons. In fact, if our nervous system had axons that were the diameter of squid giant axons, we would have brains the size of beach balls. By the way, a lot of what we know about action potentials comes from studying squid giant axons. They're so big that you can just dissect out a squid brain or a squid nerve, put it on a dish, and you can directly record from it or directly study it without really any fancy microscope equipment. So that's why a lot of what we know about action potentials comes from studying this particular type of axon. But for those of us that don't have giant axons, we instead have to have another system for preventing or reducing current leak from the axon. And the solution that mammals came up with is myelin. So myelin, we talked about already. This is the substance that surrounds some axons in the brain and the nervous system, not all. And it consists of cells, either oligodendrocytes in the brain and spinal cord or Schwann cells in the peripheral nervous system. And these cells just wrap around the axon many, many, many times with very little cytoplasm. So if you look at the cross-section of a piece of myelin like this is all just cell membrane here. And there's very little aqueous cytoplasm in between those layers, which means all the water is kind of squeezed out. And the membrane, remember, is not electrically conductive, even though the cytoplasm is. So that basically just prevents current from leaking out through the axon, and it means that the axon can be thinner for the same velocity, the same conduction velocity, than an unmyelinated axon. But one thing that's important is you can't have the entire axon myelinated, because if you did, even though the myelin prevents some current leak, it doesn't prevent all current leak. So for a long axon, if the entire thing were covered in myelin, you would the action potential would essentially die out. The signal would die out because some current would leak out. So even though it would be faster, you can't myelinate an entire axon. So instead, the way myelin is formed is it forms in these segments, and each segment usually is made up of the membrane of a single cell, and the segments are separated by these gaps called nodes of Ranvier. A node of Ranvier that's named after, I'm assuming, the person who discovered them. It's just a space where there is no myelin. They're very thin, just a few microns, and what happens here is at these gaps there are voltage-gated sodium channels. There are no sodium channels underneath the myelin. And of course, there would be no use for a myelin or sodium channels underneath the myelin because there's very little cytoplasm or extracellular fluid around the axon for the sodium to come in from. So there is no action potential under the myelin. So the action potential starts again at the cell body, and then when it gets to one of these nodes, when the current reaches one of these nodes, if it's large enough in magnitude, it will trigger the opening of voltage-gated sodium channels, which will sort of regenerate the signal. It sort of is a gain booster that gives you more current that can then travel to the next node of Ranvier. So essentially, the action potential itself does not travel through the myelin sheath. It sort of jumps from one node to another. In fact, we call this behavior saltatory conduction. The word saltatory comes from I think maybe the Latin word for dancing or jumping, because that's what it's doing. It's just jumping from one node to another. And again, that's faster than if the action potential were to just propagate all the way down the axon with no myelin. So this is why myelin is important. It allows faster conduction along, especially very long axons. So most of your peripheral axons are myelinated. Many of the axons in the brain are, especially if they have to go from one part of the brain to another. But shorter axons might have no myelin. Myelin is, for example, diseases like multiple sclerosis are caused by the immune system actually attacking the cells that produce myelin and destroying it. And that basically prevents the nervous system from communicating with the body. And so that's the function of myelin. Again, it just increases that conduction velocity. One more thing to say about the membrane of axons is that by definition, any part of the cell that has a high enough concentration of these voltage-gated sodium channels can produce action potential. So hypothetically, any cell can do this, and it's not just neurons. So we've talked about how muscles, for example, also have voltage-gated sodium channels. They can produce action potentials. In fact, that's crucial for their function. But not all cells do, and even those cells like neurons that have voltage-gated sodium channels do not have them all over the cell membrane. So really, actually, the parts of a cell that have lots of these channels we call excitable membrane. So by definition, an excitable membrane is part of a cell that has enough voltage-gated sodium channels that an action potential can be generated and propagated. So for a typical neuron, the excitable membrane starts at the axon hillock, so the place where the axon attaches to the cell body. And in fact, we call that zone the spike initiation zone, because that is where spikes or action potentials can start. So a depolarization in the cell, and later on we'll talk about what are the things that actually can cause action potentials in real cells. But whatever it is, whether it's an artificial stimulation or whatever, if that depolarization is strong enough to open voltage-gated sodium channels at the spike initiation zone, that will trigger an action potential that will travel all the way down the axon. But not all neurons have spike initiation zones at the axon hillock. So sensory neurons, and we'll talk about them a lot more later when we talk about the sensory systems, but these are those unipolar cells, which means they only have one process that comes out of the cell body, and that process is not excitable, but when it branches, the membrane that of the part that branches does have excitable membrane. And in fact, the spike initiation zone, meaning the place where action potentials typically start, is out here in the nerve ending. So this axon may go all the way out into the skin or the muscle or whatever sensory surface you're talking about, and that is where action potentials initiate. So again, something in here has a membrane, something that allows depolarization, and that in turn will so in the case of, say, a touch receptor, there may be mechanically sensitive or mechanically gated ion channels out here in the nerve ending, and so those channels being activated could be temperature sensitive, whatever the sensory system is. Those opening and letting sodium through will or can initiate an action potential, and then that action potential would travel all the way up the axon and then past the cell body into the central nervous system. So as we'll see later, these neurons have two processes, the peripheral process and the central process. And all of that process has excitable membrane. So the action potential in those cases can travel a very long distance. These axons can run almost the length of your entire body. If you're talking about those the sensory receptors in the skin of, say, your feet or your fingers. And so you have a distance of several meters, but again, because they're very fast, a typical action potential conduction velocity is about 10 meters per second. They can get across your entire body very, very quickly. All right, so that is all we need to say for now about action potentials. In the next chapter, we'll talk about what happens when the action potential gets to the axon terminal, because remember, the axon terminal is the first half of the synapse, so that's the presynaptic terminal. So the next chapter will deal with synaptic transmission.